there, back again. This is Faja Alameen for Chem 1412, and today we're going to do a experiment on chromatographic for food coloring, which is our lab number 7. What I have done is prepared and set up all of the actual lab equipment that is necessary. I don't know if you get a shot of that, but we have everything set up. We have our well set up. We have our paper that we need in order to do our project. And we have a few little items. I'll just go ahead and explain to you what all we have so that we can go ahead and get the experiment started. First of all, let me just give you a little discussion about what the actual experiment is supposed to be about. And from there, talk to you a little bit about why this experiment is done in the first place and what it actually proves. But let me get started first and tell you about all of the little supplies here so that you can see that basically these are some of the basic things that you'll need. Out of our kit here, we have blue number 1. We have blue number 2. We have our red number 3. We have red number 40. We have yellow 6. We have yellow 5. And then we have a unknown substance. This unknown substance, basically, we're not really for sure exactly what it's going to show or what it's going to prove, but we will find out exactly what's what. We have our paper, okay, which basically this is out of the kit as well. Toothpicks, of course, and we have the M&Ms that was talked about. We have some dye tablets. I didn't have the liquid form, but I have dye tablets where so that I can do the same thing with the tablets. We have a distilled water over here, our salt, the tape that they spoke about, vinegar, okay, and of course we have our beakers right here. We have the glass beaker up here, along with the well, which is what they asked us to have, stapler, scissors, and just to go into a little bit of other stuff, a couple of packs of Kool-Aid over here too, okay? So, what we're going to do in this experiment, they want us to, number 1, show the common technique for separating mixtures, and then separating these mixtures or compounds into the constitutes components. Basically, separation is based on a physical characteristic of the components, such as size and polarity. Now, what I understood from reading the material is that these colors are the polar. In other words, they resist the actual factor of water. They're polar, okay? I'm sorry, non-polar means that they resist, but polar means that they kind of blend in. And the paper is polar. So some of the actual constants of how and why it bleeds through to the paper is the experiment that we're going to do. But what it says is that the paper chromatology is a simple yet extreme effective method for determining the presence of solution in the mixtures or for separating compounds from the actual mixtures. We're using a strip of paper, and the strip of paper, of course, is what we're going to use in order to stick it inside the actual vial to see exactly how it moves. From the information is that the salt water, when you put it in salt water, it's supposed to move slowly versus faster when you just put it in just the plain water solution. Now, all of these actual things that we have here is going to allow us to do the test, whereas that basically we can get the analysis of how far how wide, and exactly some of the other details on actually RF factors that's going to allow us to see how fast it moves up the actual paper and exactly how it either spreads or don't spread. So, and all of that, let's go ahead and get busy. I will talk to you later. Bye-bye.